My name is Abby Jones, and this is my project on the independent evolution of a lysergic acid amide in Aspergillus species. Ergot alkaloids are a family of fungal metabolites that infect grains and grasses in agriculture but can be modified into useful pharmaceuticals. The most useful types of ergot alkaloids are lysergic acid derivatives. Now, there are two types of fungi that produce ergot alkaloids: the Clavicipitaceae family and Aspergillus species. The Clavicipitaceae family produces lysergic acid derivatives, but they're slow-growing and difficult to manipulate genetically. The Aspergillus species, on the other hand, is fast-growing and very easy to culture and manipulate genetically, but they have not been known to produce these lysergic acid derivatives. Ideally, this situation would be reversed, with the Aspergillus species producing the lysergic acid derivatives because they're so easy to culture. We hypothesized that there are Aspergillus species that are capable of producing lysergic acid derivatives and that we would be able to find them using a genome mining approach. To test this hypothesis, we used a genome mining approach to search for genes typical of lysergic acid producers in Aspergillus species. Doing that, we found 3 species of Aspergillus that contain the genes necessary to produce lysergic acid. So we acquired cultures of all 3 of these species and then used HPLC analysis to test them for ergot alkaloids. Following this, we grew cultures of all 3 of these species and tested them every 3 days over the course of 21 days for their ability to produce ergot alkaloids and their ability to secrete it into the liquid medium of the fungus. Also, through phylogenetic analysis, we compared the ergot alkaloid synthesis genes between the Aspergillus species and the Clavicipitaceae. Aspergillus leporus, Aspergillus homoeophyus, and Aspergillus hancockii genomes all had gene clusters of typical lysergic acid producers. HPLC analysis showed that all 3 of these species produced large quantities of lysergic acid derivative ergot alkaloids, mainly LAH. This is shown in the figure below. Our slime course showed us that Aspergillus leporus and Aspergillus homoeophyus secreted the majority of their ergot alkaloids into the liquid medium of the fungus. This trait could be very beneficial industrially because it would simplify the purification process of these useful chemicals. Aspergillus homoeophyus also shown that it could be very useful industrially due to its ability to maintain its already high concentrations of LAH over time. On the other hand, Aspergillus leporus converts most of its LAH into ergine after about 9 days. Phylogenetic analysis showed that genes used to produce lysergic acid in the Clavicipitaceae family and in Aspergillus species, such as CLO-A and DMAW, which are boxed in green below on the left, both evolved from the same common ancestor. This was shown by strong bootstrap support. On the other hand, the genes used to convert lysergic acid into LAH, such as ESO and lysergyl peptide synthetase, evolved from a different ancestor between the Aspergillus species and the Clavicipitaceae family. This shows that these 2 species evolved this ability independently. To conclude, lysergic acid derivatives were discovered for the first time in fungi outside of the Clavicipitaceae family. 2 of those fungi, Aspergillus leporus and Aspergillus homoeophyus, secreted most of their ergot alkaloids into the liquid medium of the culture. This could be incredibly useful industrially. Additionally, phylogenetics showed that the ability to produce lysergic acid evolved from a common ancestor with the Aspergillus species and the Clavicipitaceae, but each of them evolved their ability to produce LAH independently. This shows that LAH could have great ecological value since both of them evolved it. Overall, the Aspergillus species that we are working with may be useful for further study and could be useful in the production of pharmaceutically important compounds. This work was funded by the Arnold and Mabel Beckman Foundation and NIH. Special thank you to Dr. Panachone, Sanami Fabian, and Chai Steen for all of their help.